This Week in Virology, the podcast about viruses, the kind that make you sick.
Hey, Ann Arbor, how are you? It's time for This Week in Virology, episode number 158. We're recording on November 17th, 2011. Hey, everybody, I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses. TWiV is on the road again. Today we are in Ann Arbor in the state of Michigan, and we're on the campus of the University of Michigan Medical School. And fortunately with me today to help me out is my old colleague, Rich Condit.
Hi, Vincent.
What are you doing with your iPhone there?
I'm looking up to see you. I know you're going to ask me what the weather is.
What is the weather?
And I'm going to tell you that in my hometown, in Gainesville, it is now 79 degrees.
Yeah, just like it is here.
And outside here in Ann Arbor, it is 34 degrees.
Yeah, it's pretty cold.
Walking
here.
outside, I saw snowflakes.
No.
Yeah,
Serious?
Yeah. Somebody
Right.
was with
Cool.
me. I can confirm this. Snow coming out of the sky. There's a bunch of people nodding.
All right, snowing here. Well, this is the Midwest, and it is November, so it's going to be cold. Also joining us today, two guests from here at the University of Michigan. On my left, a professor in the Department of Microbiology and Immunology, somebody I've known for a long time. I've known everybody for a long time, because I'm old. Alice Telesnitsky, welcome.
Hi, Vince. Good
Thanks
to
for
be
doing
here.
this. Good to see you.
Yeah, actually, I've never heard someone do this before, but I have to say hello to my sons, Noah and Ben.
Sure.
Hi, Noah and Ben.
You can do it. You can do it. I was coming from the airport last night. I was in a taxi at 9:30, and I saw Alice standing on the corner, just leaving to go home.
Yes, and as I pointed out, I
Dedicated.
didn't have to Skype with my colleagues till,
Great.
Also
joining us from the Department of Microbiology and Immunology, someone who I have also known for a long time, Kathy Spindler.
Hi, Vincent. Hi, Rich. Thanks for coming.
Yeah, thanks for doing this with us. We're going to talk with you a little bit. We have a good show planned for you today. Before I do that, I was invited to give a seminar by the postdocs in the department, and in particular, Stefan Taube has been my host here, and he made a little model for me that I want to show you all. This is one of the most amazing things I've ever seen. This was done on a 3D printer, and it's a model of the structure of polio bound to its receptor. Isn't that cool? Now, too bad you listeners can't see this. I'll put a picture of this up. But of course, this is based on some work we did many years ago with Jim Hogel and Alastair Stephen. And my student made the receptor, the soluble form, which is shown here in blue. And we sent it to Alastair Stephen, and then they mixed it with the virus and did cryo-EM.
It's the same colors as the TWiV logo, right?
It is what was used to make the TWiV logo, exactly.
Right.
I also noticed that it is orange and blue.
Yes?
That's significant, right? That's Gators, right? That's the University of Florida. So you got it right. That's good.
an alternate point of view would be that Michigan is blue and maize, and University of Nebraska is red. It sort of looks red and blue, and we're having that game this weekend.
All right. This is pretty amazing.
That's beautiful. So this 3D printer technology is amazing. Anyone else here ever played with this? This is amazing. You can make anything you have a PDB file for or a VRML file you can print out. So this is useful for finding out what molecules look like. And so that's one of my favorites, if not my favorite. So thank you for giving that to me. We don't want to break it here because this is also the TWiV logo. There you go. So how many people here in the audience listen to TWiV? Raise your hands. Not bad. It's not bad at all.
Impressive.
What's that?
I think it's impressive.
Yeah, it's pretty good.
Have all of you filled out the listener survey?
Yes. Thank you. Yeah, so before we start, we here on TWiV, we'd like to do some things in the future that require a little financial support. So we're trying to raise that by advertising. And we have a few advertisers lined up, and they would like to know who our listeners are. And so to find out, we'd like you to fill out this listener survey, which is at triplemojo.com/TWiV. So we'll put a link on the website. It'll take you about 6 or 7 minutes. and it will give them demographics that we can use going forward. So we're going to keep repeating this for a few weeks until we get some good numbers. So please do that.
How many people have done it so far?
1.
No, I mean, out there in TWiV land.
The last time I looked was just under 200.
Right.
Now, TWiV is regularly listened to by up to 10,000 people. So if you want to help us, this is the way to do it. Fill out this survey. We don't want to have money from you. We just want to know what advertisers we can get to support the show. We have a lot of good plans for TWiV, and to do that, we need a little financial support.
They're going to double my salary.
That's right. Triple.
My TWiV salary.
Triple your TWiV salary. So let's start talking with our guests here, but find out first where you guys came from. Alice, I know you were a postdoc in the golf lab because I'm right next door, and for a long time I saw you on a daily basis. But before that, where did you begin? When you were haploid, as Rich would say.
I did love that. That was absolutely lovely. I grew up in California. I was born and raised there. Something like 5th generation Californian, pre-gold rush family. And I went from kindergarten through PhD in California public schools. So I got my undergraduate degree at UC Davis, and then I went to UC Berkeley for my PhD.
What did you work on?
I worked on E. coli RNA polymerase transcriptional elongation.
Wow. There you go.
And
I have so many questions today.
Alice and I actually had found a lot of common points in our history today. Yeah.
we worked with a lot of the same people. We knew the same jokes, things like that.
That's right.
And then I came here. I got a job. Actually, when I was interviewing with Mike Imperiale, actually what happened is Mike called me up for a faculty interview, and he said, we need to have you come as soon as possible. And at that point, I was just about 10 days overdue with my second child. And so he said, we really need you to, or maybe it was it was just a week before I was due. And I said, well, I have to, put it off because I have this poster at Cold Spring Harbor that I'm giving or something. And I wanted to put it off. And then at that point, yes, exactly, it was a couple days before I was supposed to go. So I had my son induced, and now he's 18, and so I've been here for 17 years.
And you love it here, right?
Actually, I've come to love it here. The weather is. I am a horizontal farmer here. Many people in the audience ate vegetables from my garden all year round, and or all through the summer. And I get to teach some fun classes, and I have had superb graduate students and postdocs. And you're going to get to meet my postdoc at dinner, I guess.
Wonderful. How about you, Kathy?
So I grew up in Ohio. I went to undergraduate at Purdue, and then I moved to California. I did my graduate work at UCSD. I started out working in phage biology with Masaki Hayashi, worked on Phi-X174. And then partway through my PhD, I changed advisors and projects, and I started working with John Holland, and I worked on vesicular stomatitis virus and persistence. And then I moved up to UCLA for my postdoc with Arnie Burke, where I worked on adenovirus. And then I took a faculty position at the University of Georgia in the Department of Genetics. So I was in Athens for 16 years. And then Mike Imperiale was instrumental in talking to me about the job that was going to be open here. And I came here almost 10 years ago now. So
And you love it here as well.
I do like it here. It's pretty good.
Good job, Mike.
And actually, a thing for your listeners to keep in mind is that we are just 3 of the fabulous virology faculty here at the University of Michigan. It's an excellent place to do graduate school.
That's right. And I'm impressed. So Rich and I are spending these 2 days talking to a lot of the faculty, and it's great. You're very lucky to have a lot of, not just virology, but other outstanding faculty as well.
Yeah, I was gonna mention at the start of my seminar, after poking around here for a day, this is really a great place to do science. And it's a really high concentration of virologists. It's a good virology faculty.
And coming from a department where we only have 2 virologists now, I really appreciate having more. It's important to have people who think like you do around you. Also think different, but also think a lot like you. So good job for hiring all these good people. So let's start with Kathy over there. And I want to talk about mouse adenovirus, which I know you've worked on for a good part of your career.
Mm-hmm.
And I want to know where this virus came from originally that you work with.
Okay, so it came out of someone's freezer, if you want to be really technical. Wally Rowe Hartley and Rowe is the first paper describing mouse adenovirus, and they were trying to purify friend leukemia virus, and they were getting cytopathic effect that wasn't characteristic of the leukemia virus, and they pursued that and purified the virus, and found that it had some similarities, mostly serological, with human adenoviruses, and then not a whole lot was done on it for a long time.
When would this have been?
So 1960 is the original paper description. And then there was a second adenovirus that was described in 1967. And then very recently, a mouse adenovirus type 3 was described. And interestingly, it was also isolated when somebody was trying to purify, in this case, a hantavirus. And they got this cytopathic effect and pursued that, and it was another mouse adenovirus.
Even the human adenovirus was a culture contaminant, wasn't it? If I got that right?
My understanding was that it was isolated by two different groups from, I thought, respiratory infections.
Right, I think they're trying to grow tonsil tissue or something
Well,
like that.
right.
Yeah, right.
So, yeah.
Okay.
So at what point did someone call it a mouse adenovirus? How did they find that out, that it wasn't something else?
Pretty much it was Hartley and Rowe in that 1960 paper proposed that it was probably an adenovirus based on the serology. So it cross-reacted with serum to human adenovirus.
So have people looked in mouse populations in the wild and looked at the distribution of this virus?
So not a lot is known about that. There have been a few isolated studies, some studies looking in Australia. We don't really know the natural prevalence of the virus in the wild.
But it is out there. We know that similar
It is out there, and certainly that's the case for mouse adenovirus 2, and then this one that was isolated from this striped, some kind of field mouse, the mouse adenovirus 3. And I don't know that there's really much evidence for where mouse adeno 1 is in the wild, and MAV1 is the one that we've been working on.
Is that something that is worth doing?
For somebody, perhaps.
Yeah, not you. I'm not saying.
Right.
How about in animal rooms? Is it a problem?
So most of the mouse companies test for mouse adenovirus, and colonies are clean. And so no, there's not a lot of evidence for it in mouse populations.
So in terms of the virion structure, the genetic organization, it's all very similar to human adenoviruses?
It is. There are a few differences when you start looking, at the finer detail level, in terms of actual transcription units, and in some cases different genes are encoded and so on. But by and large, yes, it smells, looks like everything. It's an adeno.
So what I always found compelling about your work from the beginning was that, if I'm correct, your inspiration was working on a natural host pathogen pair, right? Can you elaborate on that?
Right. So I can remember specifically in my postdoc walking back from a pizza birthday lunch and talking to Arnie, my postdoc advisor, and we were both kind of lamenting the fact that there wasn't really a good animal model for adenovirus, and if somebody could just do that. And so I started looking into that, and Arnie was very generous in allowing me to start working on mouse adeno while I was still in his lab.
And so I remember some of your first experiments were mouse blots.
Right.
Right?
Right.
And it occurred to me...
What do you mean by mouse blots?
I'm going to have her describe this. So you must have gotten this from Luis Villarreal, is that right?
Exactly. So mouse blots, and actually Ashley Haas has, I think, done sheep blots. So you don't have to just do it on small organisms, but you can imagine the size of nitrocellulose you need for a sheep blot. But mouse blots, you embed the mouse in a freezing medium, and then you section down through it with a microtome, and then you take a piece of nitrocellulose and put it onto that and make another slice, and then you have your section on the blot, and then you treat it like a Southern blot. You denature it and probe with a viral probe. We used P32 probes. And then from that, you can tell where the virus is. And so, yes, in fact, I went up to UC Irvine to learn how to do mouse blots.
Because I think Luis really was the father of that technique. He did it with polyomavirus, as I recall.
Right. And I learned it from Rosemary Rochford, who used to be here at Michigan, but I learned it when she was still at Irvine.
So when you do that with a mouse infected with mouse ad, what do you see?
So you see a lot of staining in the spleen and also in the brain. And those are the places where we find the highest levels of virus when we look by other mechanisms also. But essentially, if you wait long enough, you see virus all through the animal. So it's really a pantropic virus.
So these are done in laboratory mice, right?
Mm-hmm. Mm-hmm.
Do we have any sense of whether it would be different in a wild mouse at all?
That we don't know because we just don't know that much about infections of wild mice.
And we don't even know the pathogenesis in wild mice, right?
Not really.
And you can't bring wild mice into our mouse facilities. People would freak out, so you can't do those experiments. You can do them at home, I suppose.
Right. Maybe not at my
So when
house.
you inoculate your mice, you probably do it IP or IM or IV, something like that, right?
We do it intraperitoneally. It's a pretty easy way to do it for a molecular biologist.
Yes, and in the wild, we have no idea how it would be spread in mouse populations.
Right. One of the things we looked at, and also Hartley and Rowe in their initial paper, was how is the virus disseminated from mouse to mouse? And it doesn't appear to be by aerosols. You can put mice in the same room as infected mice, and the naive mice will not get infected. We've even put uninfected mice on infected cage bedding, thinking it might be transmitted by fomites, and it's not. So only when we put mice together, and we did that with male mice, and so of course they're very likely going to fight at that point. And so then we got some transmission, and so about all we can postulate is biting, direct contact, biting, kicking, scratching.
So is the virus in respiratory secretions, urine, feces?
It's secreted in the urine for sure. I don't know that we've ever found evidence for it in the feces, but I think others have. Respiratory secretions, if you infect by a respiratory route, then I think you'll find it in respiratory secretions, but whether that's a natural way, I don't know.
So when you infect a mouse in the lab with AD, is it typically an acute infection, or are there persistent infections as well?
So mouse adenovirus does cause both persistent and acute infections. Most of our experiments that we've been doing in the last 10 or so years have been looking at acute infections, but in the past we have done experiments to see whether the virus persists. And our best guess is that maybe it's persisting in the kidney or the brain. We know it's shed in the urine for more than a year after infection.
And those mice have no symptoms during that time?
Correct.
Do you understand how the virus can persist?
No. Good question.
So in an acute infection, do the mice typically die, or do they survive?
So it depends. For the mice, it depends on the virus and the dose. So whether it's a wild-type virus or a mutant virus and the dose, and it depends on the virus strain. It also depends, to some extent, on how old they are. There's a little bit of age dependence if you gave the same dose to it.
So it depends on the mouse strain.
Right.
Right, okay.
Right. But if you gave the same dose of virus to a neonate and an adult mouse, you would probably see some differences as well.
So a bit of your work deals with characterizing the replication in macrophages. And what was the reason for doing that?
So we had some evidence that the virus infects both endothelial cells and cells of the monocyte lineage, so macrophages. And because macrophages are one of the innate immune response cell types, it occurred to us that maybe the virus is in a way sort of analogous to HIV, where it's infecting a lymphoid cell, sort of taking down the immune system, that maybe the virus is really doing something that attacks the macrophage ability, or it could just be using the macrophage as a place to replicate, and maybe the macrophages can help overcome the infection. And so our experiments kind of gave us both answers. It seems like the macrophage is a target, but it's also an effector against the viral infection.
So the virus can replicate in macrophages?
Yes.
So if you deplete macrophages, do you impact the spread of the virus?
Right. So we've done experiments where you treat with claudronate liposomes, and that is a kind of a drug-like effect that does deplete the mice of macrophages. And when we look at virus levels in the brain, there's really no difference upon depleting the macrophages. But if we look at the spleen, a strain of mice that's ordinarily ordinarily resistant to the virus then becomes more susceptible after depletion of the macrophages. So that's our evidence.
So you're going to tell us this, but then, I mean, it's interesting that you don't have any less in the brain, so how did the virus get into the brain?
That's another good question. I'd like to have people come and help us try and figure that out.
You don't know how the virus gets in, whether it's free virus or within macrophages.
Exactly.
Right?
And
she said if she depleted the macrophages,
it still gets in.
Right.
Well, it could be both ways, right?
Right.
So if you deplete macrophages and the virus still gets in the brain, how does it do that, do you know?
That's, again, we don't know whether it's, just free in the bloodstream and then, lots of questions.
You've also done some work where you characterize the breakdown of this so-called blood-brain barrier by the virus. Mm-hmm. Could that contribute to entry of the virus?
So it probably contributes to how the virus is getting into the brain, although we don't see the virus replicating broadly in the brain parenchyma. It just seems to replicate right around the blood vessels. So, as I mentioned, it infects endothelial cells, which are the cells that line the blood vessels. And we don't know how the virus gets into those cells. So we don't know anything about the receptor for the cells in vivo. But in terms of the virus breaking down the blood-brain barrier, this is a barrier that, everybody's heard the blood-brain barrier, and then when you start to find out more about what's involved, it's tight junctions between these endothelial cells, and then there are contributions from accessory cells, notably astrocytes. And all of this goes together to form a barrier that doesn't even let molecules through that are any bigger than 500 molecular weight. So it's a very tight system. And there are a number of viruses that can break down the blood-brain barrier. HIV, West Nile virus are some of the ones that are the best studied. And at least for West Nile virus and HIV, the thought is that the virus breaks down the blood-brain barrier in part by getting in through the endothelial cells by a Trojan horse mechanism. So the idea of infected monocytes traveling either between two endothelial cells or right through the middle of an endothelial cell, which is really kind of a strange concept. But we don't really have any evidence for whether mouse adeno does that. Other ways that a lot of viruses do things to break down the blood-brain barrier are in part by inducing inflammation, which can bring about chemokines and cytokines and things that can start to disrupt these tight junctions. And a lot of the cells in this neurovascular unit can express matrix metalloproteinases, or MMPs. And those enzymes are known to degrade tight junction proteins and also basement membrane that is also part of the function of the blood-brain barrier.
So I have this really naive perception of the blood-brain barrier. The first time anybody ever mentioned this, I envisioned some sort of sieve or something in my neck, okay, that was making sure that nothing got to my brain, right?
Okay.
But this is what we're talking about, is vessels in the permeability, if you like,
right,
of the blood vessels in your brain.
Right.
So this is the idea is to, on an evolutionary scale, to protect my brain. It's a privileged site. Is that the idea?
Right. It's privileged from lots of things that would get into other organs in that kind of sense. It's not totally privileged in the immune sense. So a lot of people think of, immune cells don't get into the brain, and that's not really the case. But there is this protection. As I said, molecules larger than about 500 Daltons don't get into the brain, and that is a serious problem for trying to introduce
So the structure of the blood vessels in the brain is fundamentally different from elsewhere in the body?
Yes.
And tight junctions is a big piece of this?
Exactly.
Okay.
Right. Endothelial cells form tight junctions elsewhere in the body, but in the brain they're very unique, in part because of these accessory cells, the astrocytes, and some of the other cells.
And ordinarily this would keep viruses out, but mouse adenovirus has found a workaround.
Right.
And what is the workaround?
Well, that's what we're trying to figure out. We have some evidence that the virus induces high levels of some of these MMPs, and we see increased MMP activity both in vivo and in some of these brain cell types in culture. But then the question just goes one step back. Well, how are they inducing this increased activity in MMPs? Is it a transcriptional thing? Is it some kind of response to reactive oxygen species that the virus has induced, or chemokines or cytokines, things like that? So.
Now, I know I'm kind of anthropomorphizing this, and this is a big why question, and we're not supposed to ask why questions in biology, right? But why would a virus want to do this? I mean, it can infect other tissues and that kind of stuff. You're not going to spread from the brain, I don't think.
How do you know it wants to do it?
Well, that's why I said this is a stupid question, okay? But I'm into stupid questions.
Right. So that is a good question. I don't think we have the answer for that. Why do viruses do a lot of the things they do?
Okay.
Maybe an accident. It really wants to grow somewhere else,
yeah.
right? Not many viruses get in the brain. It's pretty well protected because of these junctions. But then once you do get in, it's a mess. It's a big problem.
Right.
Now, did you say the virus replicates in the endothelial cells?
It does replicate in endothelial cells, definitely in culture, and we've seen evidence by in situ hybridization.
Is it lytic? Does it destroy those cells?
I don't know that we have evidence for that in either case being totally lytic, but just by analogy, we've pretty much always assumed that,
that's a simple way. It would just get in by destroying the endothelium, and then the parenchyma is right there. Right. The other thing I wanted to talk with you about is your characterization of genes in the mouse that confer susceptibility or resistance. So you've done a lot of work on this.
Right.
Just give us a general picture of how you map such genes.
The big picture.
The big picture.
So we started out a long time ago noticing that there were differences between different strains of mice in their susceptibility. And so we did classical genetics, crossed a resistant strain with a susceptible strain, took the progeny from that, and then did back crosses. And the progeny from those back crosses then were mapped with respect to what their genes were like. Were they like the resistant parent, or were they like the susceptible parent? And we did that all at the DNA level with PCR-based microsatellite markers. Then
So did you do all those crosses and stuff yourself?
We did.
That's a lot of mice.
It was a lot of mice.
Okay. How many? So these are recombinant inbred strains.
They're not recombinant inbreds. They're just back-cross mice. Right.
Okay.
Right. So we take inbred strains and cross those, and then take the progeny from those and back-cross them to one parent type.
Okay. So, but you wind up identifying a number of different strains with different susceptibilties, right?
Well,
each
individual mouse then.
Okay.
And then those mice were tested for their susceptibility to the virus. So we infected them and then harvested them, measured their susceptibility by a virus load in the brain type of assay. And then we took their DNA and genotyped it at markers across all the genome.
Okay.
And then put the genotype and phenotype data together with the help of somebody who knows how to do that sophisticated computer programming, and got an answer that susceptibility, a major locus for susceptibility, maps to chromosome 15. And then we did additional crosses and genetic things to narrow the region. And so now we're at about 750,000 base pairs of DNA and about 14 genes.
Okay, and what are those genes?
So these genes are the Ly6 genes, and it's sort of a good news, bad news thing. They're a gene family. And the good news is that it's a gene family that makes sense from the standpoint that they're expressed in myeloid and lymphoid cells. They're induced by interferon. They're found on the cell surface. The bad news is that they're a gene family, and so it makes the region really difficult to kind of tease apart. But in a way, that's a good news thing, too, because when susceptibility has been identified in other microbial systems, it always seems to map to a gene family, as if having a mutation in one of the genes still allows other genes that might have redundant functions to work, but this particular microbe has figured out one particular one to act on.
So is this in these experiments, is this the only susceptibility locus that you came up with? I mean, there might be multiple loci, right?
Right. In fact, it's called quantitative trait locus mapping, QTLs, and we did find others, but this was the major one. Okay. And there are others that, if we had more time and money and mice, we could go after. But
so how are you going to narrow this down to one gene?
So one of the approaches that we're using is basically a marker rescue type of experiment. So we've made transgenic mice where we've taken the interval of DNA from a susceptible strain and introduced it into, or we're introducing it on a bacterial artificial chromosome, into a resistant strain. And the susceptibility is dominant, so if we get progeny mice from that are susceptible, then it means that that piece of DNA carried the gene on it. So,
does it mean anything to you that the susceptibility is dominant?
That's a question we get a lot. It seems to be hard for people to get their head around the fact that susceptibility might be dominant, but it doesn't really give us any clues or worries.
Does that mean that the resistant animals lack the protein, for example?
It could be, but there are a lot of biological mechanisms that you could postulate.
So is it your plan to narrow it to one gene and then try and figure out the mechanism?
That would be our dream, and I think my graduate student, Tian, who's working on it now, maybe thought that would happen by the end of her PhD, but maybe it's going to need to go beyond that. And it may not be just one gene in that interval. It might be more than one gene.
But if you narrowed it down to one gene, so then you would take that and compare it in the resistant and susceptible mice, and you would see some difference that would explain the phenotype, right?
Right. So a lot of times people ask us, can we assay for susceptibility in cell culture? And I always say, well, when we know what the gene is and its function, then yeah, we'll be able to do some assay in cell culture that shows us this.
So how about you have virus that can grow out in the mice that aren't ordinarily susceptible, sort of resistant viruses that would give you a clue of what viral protein's interacting?
Yeah, operationally that would be pretty hard to do, though, I think. But theoretically, you could think about doing that.
So what's your advice to new students or postdocs who want to work on mouse models for virus diseases?
So that's a good question, because I think mouse models are really important, or animal models in general. They tell us things or allow us to do things that we can't do in humans. For example, adenovirus 14 is a virus that's been pretty virulent in the United States, and I asked my colleague just the other day, well, what do we know about why it's more virulent? And she said, well, we're kind of limited because there aren't good animal models to test that. So animal models can help you a lot. Wherever you can kind of bring it into things that are relevant to the human virus in this case, or in our case, we've kind of diverged because although mouse adenovirus has similarities to human adenovirus, particularly in persistence and it being a problem for immunocompromised individuals, it doesn't seem to have the same kind of upper respiratory infection, and human adenovirus, for the most part, isn't found in the brain. But we're taking advantage of the fact that we now have a mouse virus, a natural mouse virus that causes encephalitis in the brain, and we can take a look at various innate and innate immunity and biological things that haven't been explored.
I'm a big fan of mouse models, of course. The problem is we have to convince the public, and there's problems like there's an article I'm looking at slate.com called The Mousetrap, The Dangers of Using Lab Animal to Study Disease. And so the public will read this and think, we shouldn't be supporting this kind of research, but it's our job to counter that with intelligent reasons to use this.
I think especially for mice, it's very important to have animal models because sort of similarly, I think you're probably aware that there's a move afoot now to totally outlaw the use of chimps in research, for example. And so the main disease in the virus system that I study, HIV, we're really limited that we can't test drugs in an animal, and the development of animal models there. You should have Theodora on at some point. It's very interesting to try and chip away at the blocks there. But so I think that this work that Kathy's doing is hugely important just to sort of verify those approaches. And it's not just, ethical things are hugely important in what society thinks, but I think just in terms of the cost and the benefits to people to be able to trust results, you can't really just test things in a Petri plate or whatever like this. So Kathy's work is hugely important.
So Alice, let's move to you and talk about what you do. And one of the things that you work on that I found interesting is you're studying the sequences in the HIV genome that direct dimerization and packaging of the RNA. So of all the things you could have worked on, why did you start working on that? What's the interest?
Okay, and in fact, the direct reason was I got paid to do it. Hey, some of us have no shame. But so in fact, it's interesting then that I suspected that you would ask me after you asked Kathy to talk, because we did have that nice paper in Science just a month ago.
Right.
But the real creative part on that project was done by my collaborator. And the hard work was done by people in my lab. My postdoc, Sergei Harasonchik, out there, he did really the hard work, and some of setting the system up was done by a previous graduate student, Eric Garcia. So we got into that, and that was a biophysical coup. So I'll tell you why it was in Science. What was really creative about it was that RNA structures are hard to study because they're hugely charged molecules, and they're sort of the same all over the place. So people haven't been able to solve the structures of really large molecules. My collaborator, who's Mike Summers at UMBC, is an NMR structural biologist, and he introduced an innovation, which is basically he sees that the interaction of adenosine residues, when they're present in AU base pairs that are adjacent to one another in a structure of RNA, give a sort of signal in his NMR so that he can localize these interactions inside of a larger structure. And basically then there was this technological breakthrough by introducing just one of those kind of situations within a molecule, where he can now look at RNAs that are 10 times as large as any RNAs could ever be visualized before. So that really, the reason that paper was in Science was because of that technological breakthrough. But we're also addressing a really important biologic question, which is how HIV selects its RNA from a milieu of all the very similar looking RNAs in its cell. So that was the part that we sort of got hooked into, is that we wanted to validate that the things that we were testing structurally that were leading to models for how the virus would recognize its RNA were in fact valid. So we were designing ways to test whether or not changes in the RNA that stabilized alternate folds had the predicted replication outcomes. And another thing that was tricky is, of course, the virus, well, actually HIV is a hugely small virus. It's only like 10 kilobases long. And this region that's been mapped to be involved in the specific recruitment of genomes has been known for 30 years and localized forever to this like 500 base region. And it wasn't always HIV, but all retrovirus is sort of the same. But people have debated for 30 years or more what really is important for the recruitment of the RNA. And the problem is that when they make changes, it will frequently destroy this ability to recognize the RNA. And so then that would seem to imply that that region is important for recognition. But other pieces of that same region would seem to be sufficient. So there would be big debates in the field. And as we start gaining a structural understanding of this RNA, all of those sort of conflicts make sense. So that, for example, things we now think that we do know something that is sufficient. But when changes are made outside, sometimes they can affect the ability of the structural feature to play its role. And this gets back, especially in nucleic acids, to what I told you, that any regions can base pair with any other. So of course, I can't show you like pictures that have stem loops, but you guys are all sort of familiar with the stem loops that form in RNA and their hydrogen bonding between localized sequences. And if we looked at the landscape, it would be like a bunch of different trees all sort of popping up on the landscape. If we then took the horizon and sort of circularized it around and to make it into a globe, there'd be trees all sort of sticking off the side of it. And if the center part, the globe, was what was important for packaging, we could lop off all the trees and the globe would still work. But if you just sort of messed up one arm of one of the trees, because we know these are forming hydrogen bonds between them, then that sort of displaced other arm is going to want to do something, and it's maybe going to jab into that sphere that's in the middle and make the sphere no longer nice and smooth and round and able to interact with the packaging machinery. So this is the sort of thing that we could go back and sort of retrofit our understanding of the structure to problems that people had in the past. And some of the things that are really, I think, quite fun about the structure then is, and again, I told you earlier, because I was in the Goff lab and we had this birthday party for Steve last weekend, I was having fun drawing an analogy to, these guys might be able to see this, Jack-in-the-Box. You remember this Jack-in-the-Box sort of thing? So the sort of thing was, again, and I'm showing a hairpin here, we cut off one arm of it and it prevents poor Jack from jumping out of the box. And in replication, we need to have both the phases. We need to have sort of the portable phase where Jack is stuffed into the box, and then we have to be able to allow Jack to pop out of the box at the right point. And some of the trickier parts of the interpretation is that we have to make sure that the lid to the box doesn't keep pushing Jack back in there and so forth. And these things you wouldn't really understand until you sort of had a understanding of the structure that you're working on. So I think that actually this was hugely exciting from a biophysical point of view, what we published. But the biology part's still to come, and I can tell you about that too.
Yeah, so before you do that, so my understanding from the science paper is you have a small region of the RNA that's involved in multiple functions. It's involved in dimerization and packaging, putting the RNA into the capsid, and translation as well, right?
Yes, and even more, and even more. So some of these things, that, like when we had Rich's seminar this morning, and we're talking about pausing and so forth, there's also RNA structures that are involved, are required for an escape from initial, what we might call abortive transcription in bacterial system and elongation. So this is another issue as well. And some of the things that my postdoc really did a masterful job is when we start messing with one part of that RNA that, for example, limits transcription, we're not going to see as much of the RNA, and sometimes then there'll be concentration effects that will affect the outcome. So we had to work rather hard to try and minimize the variables. So we, despite the fact that the transcription was lower, we had to work very carefully to make the other one have the same low transcription. So everything, making all else be constant. But yes, exactly.
So I'm going to ask a why question. Is that okay?
It's okay.
Why are these three and more, you say, functions all within a short region?
So why they're all in this short
Yeah,
region.
yeah.
And there are probably other RNA, well, in fact, actually, Steve Goff and another collaborator have a paper coming out shortly about another RNA motif that's in a protein coding region, but to some extent it's because a lot of the genome is packed with coding sequences, and there is a little bit of plasticity in the coding region. But so this is the 5' end where it's non-coding, that those are all packed in. So in fact, but it does, in fact, one of the regulatory switches that was, in fact, sort of the critical key that we unlocked in this paper, the biologic thing, does overlap with the start codon, and that's why there's been some confusion about whether or not the packaging signal extended into the GAG region. And in fact, what that is when you start messing around out there, you force the RNA into alternate folds that damage the necessary motifs.
Mm-hmm. So before you can package, the RNA has to dimerize, is that correct?
That's certainly what we think in general, and it's sort of the dogma. And so in fact, what people say is that dimerization and packaging, at least those physically on the genome, they are so closely linked that we think that they need to go together.
The dimerization exposes something that's needed for the packaging step.
And so, and in fact, actually, it's interesting. So now we're jumping ahead to the next paper that we hope to submit shortly with our collaborator. But the mechanisms of exposing regions that are necessary for specific interactions between this RNA and the viral proteins appear to be different for HIV and for our favorite gamma retrovirus, murine leukemia virus. So in the gamma retrovirus, there's a more severe sort of rearrangement in exposure of the high affinity binding sites. And for HIV, it's, in fact, actually, the RNA monomer can fold in two different ways, one that's dimerization competent and one that isn't dimerization competent. So the dimerization competent monomer contains the high affinity binding sites for the proteins that get it in. And then there are some structural changes that happen after dimerization occurs, but that wasn't the question that you asked.
So, all right, so then packaging occurs, which is the interaction of this structure with nucleocapsid protein.
Right, and of course at that point it's in a polyprotein. I first heard about polyproteins from
polio, right? Exactly. Yeah, it was the first polyprotein discovered by Don Summers, a long time ago.
Wow.
Right, and that's where you learned in some virology class. Right?
No, actually, I learned that during my grad school interviews with Eckhart Wimmer.
Okay, that's fine. No problem. So it binds, the RNA binds to this polyprotein, which then goes to the membrane as part of the...
That's correct, and this is there's a few things. So I tell you, that was the creativity on this project came really from our collaborator, but it starts tying into questions that I'm interested in my lab too. And of course, sometimes you become influenced by your environment, so we're getting more involved in this dimerization and packaging. But a big question for me has frequently been, why do retroviruses encapsulate only a single dimer of RNA? Mm-hmm. Why do they encapsulate pretty much precisely a dimer of RNA? And so
You asked the why question.
Yeah, I was going to ask. I'm glad you did.
So, and in fact, so then this is the point, as you say. But at first I was railing on the whys too. But right. At any rate, but the point would be, so we'd say like, what is the mechanism by which the RNA forms some high affinity interactions? And through our collaboration with Mike Summers, we think that at least in the case of Maloney murine leukemia virus, there's about 12 high affinity interactions between GAG and the RNA. However, we think that there are 2,000, 5,000 GAGs that come together to form a virion. And we know we overexpress the RNA, and except for some little tricks that Silas can do, we generally don't get extra RNA in the virion. So somehow the virus, after it's made these 12 high affinity interactions, the other molecules which are capable of doing those interactions are somehow educated to no longer do this. And it's a really interesting question.
Is there just no room? Is that?
No. In fact, so that's another interesting thing that I did when I first joined my lab, is I made genomes that were 3 times the normal length. And so they dimerized, and they packaged fine. In fact, Ela Singh did some structural work on those, and she never published it, but what she saw is the virions formed, but then they had long tails coming off. So extra GAG got recruited to make a particle that could accommodate those long genomes, and it still had the same curvature in the top, but it was basically assembly was nucleated by a dimer linkage, and then it grabbed all the RNA that was covalently attached.
So let me get this straight. A polyprotein binds the RNA. That polyprotein is GAG, Pol.
So that's the other thing we were talking about, and this is, again, getting back to fun things we're doing in our lab, and that you made me, because I had to talk to you this morning, sorry about that. When we were in the midst of a very exciting data club, and I had to come out and talk to you about,
I'm really sorry.
Okay, but at any rate, the point would be that, so GAG is like 65 kilodaltons, and then, as you know, there is this, for retroviruses, 95% of the time, translation terminates at the end of the GAG.
Right.
Okay, and 5% of the time, translation proceeds either by frame shifting or stop codon suppression, and you get this in case of MLV GAG pol, which Silas tells me is 180 kilodaltons. So this thing is like 3 times as long, and you can imagine then, as we were pointing out, that this basically becomes linear, and it's sort of like the radius of a forming particle. And unless you made a particle that had a radius that was 3 times as long, you wouldn't be able to pack in all those GAG poles. And this was something that Steve Goff had shown a long time ago with Felsenstein and Goff in like late 80s, 88 was it? That in fact, that you could not assemble a particle if you express just GAG pol. So you can't form a particle with just GAG, and this 5% of the pol, which is twice as long as GAG itself, we think can then be accommodated in assembling particle.
So can you are the particles you would make without any RNA exactly the same as those with RNA?
So according to my friend Paul Bienash, they bud with the same kinetics, but are they exactly the same? I don't know, but pretty much the GAG ones are pretty much the same. You would agree, and without genome. And so it's actually one of those interesting things. I tell you they need exactly one genome, but they can form with no genome at all, and that's the basis of, in fact, retroviral vectors that we use all the time. So this brings me to what I do love to talk about.
Sure.
So so this was
if so far you didn't like anything that we said.
I was just going to jump into the things
okay.
that jazz us. I mean, not that those things don't sort of excite us, because science is so cool. I mean, this is when, if we all had all the funding we would be up till midnight every night just reading papers in Journal of Virology and getting excited. Mm-hmm. And you do that as an editor of the Journal of Virology.
Of course.
Get excited. But the rest of us have to work. So the thing that we've been excited about for some time is that we know that retroviruses require RNA for assembly. We know they don't require their own genome for assembly.
They require RNA for assembly?
In vitro, and...
But I thought you said you can make particles without
So therein
RNA.
lies the interesting point, is that retroviruses do not need their genomes to assemble, but they need RNA to assemble.
Okay.
So therefore, if they don't need their genomes, but they need RNA, what do they use?
Something from the cell, I guess.
Whoa, very good. So then the point was, so we then reasoned then if they need something from the cell, and then if we looked and saw what was there, that would be what they needed. Does that seem to make sense?
Makes sense, yeah.
So it does. So I've been kind of playing with this since my very first graduate student, Nicole Robson, but just kind of in little ways. But the bulk of the work was done by a former student, Adewumi Onafo-Anuga, who did the work that Kathy Collins described to you, because she's now a postdoc with her. And then my current student, Sarah Keene, is doing a lot of the work here. So we're studying these RNAs that are in retrovirus particles. And they are really fascinating. And the main one that's there, we don't know whether or not it's necessary for assembly because it's necessary to cells, so we can't knock it down. But we have learned a lot about it makes multiple different interactions with GAG. It's called 7SL RNA. It's the scaffold of signal recognition particles. And it seems, teleologically, whatever the right word is, to be a good candidate to do these sorts of things, because what this RNA does in cells is it remodels proteins in a way that it catalyzes reactions. So Peter Walter says it's an enzyme itself, and it's amazing. And probably it's not necessarily appropriate for TWiV to go through all of these fascinating activities, but there's this RNA, it binds proteins. What it has to do is recognize, so it's an RNP with the RNA and protein, sort of like a ribosome, but it then will recognize the signal sequence on nascent polypeptides and take those secreted proteins to the endoplasmic reticulum. But what's pretty fun is a couple things that happens then. What happens on the signal recognition particle is it recognizes the sort of greasy leader peptide that's coming, a nascent peptide. And once that peptide interacts with SRP, it causes a conformational change in the RNA. Okay, but then once the RNA has changed shape, it actually changes the affinity of the signal recognition particle for its receptor. So then, again, Peter Walter would say it's an enzyme, because at that point, the affinity is changed. It has 100-fold higher affinity. So that's why there's regulated trafficking to the ER. Once it gets there, then the RNA will introduce a pair of GTPases and change their shape so they're activated without any GTPase activating enzymes. So that, in fact, the RNA changes the shapes of the proteins to make them into active GTPases. So what this RNA does professionally is meet and greet and change shapes and so forth. So it's really sort of an architect. So this is why we're kind of excited about it. But we can't tell whether or not it's important for viral assembly. And that's sort of simplified because we have some clues on it. But I just want to tell you one story that you did read, which was about the yRNA. So then we're interested in what are the RNAs, and one of them was this 7SL that I just told you about, and it's present in like 12 copies per virion, so molar excess over the genome. There was another RNA that I discovered in there, sort of by accident, and this was using the MLV system. And so then here suddenly, this RNA, which is called yRNA, I thought it was an opportunity to see whether or not this RNA is necessary, because unlike signal recognition particle, which is essential for cells, people don't really know what yRNA does, and it's part of a complex called RhoRNP, and that's the major autoantigen in lupus. So people who get lupus, they have antibodies to this. And a collaborator knocked down the Rho protein, which usually forms a complex with this yRNA, and we have a functional knockdown of the yRNA, because if we knock out the protein, the RNA no longer is present, because the RNA is born and dies in the nucleus in that case. As soon as it's transcribed, it usually joins up with the Rho protein. Because we had an actual Rho knockout, there was no Rho protein, and the RNA was instantly degraded. But then we tried to see whether this RNA that's usually abundant in viruses was necessary for virus replication by letting the virus replicate through those cells. And we were a bit disappointed that the virus replicated and ran through them at a fine rate, but the thing that was interesting to us then is we looked in the virus particles, and they were still chock full of this RNA. So this RNA that's born and dies in the nucleus, and we usually think of retroviruses as assembling at the plasma membrane, had enriched itself for this small host RNA, many-fold, tens of fold higher enrichment than the genome itself. So you think in terms of picking out a straw from a needle from a haystack. So it really suggests that the virus could not escape incorporating this RNA, even though the RNA is nowhere near the site of assembly.
Do you know where in the particle the RNA is binding?
So we don't know that, and those are certainly things we would like to do.
Because if you could, you might be able to alter that and see if you could disrupt it, and what would happen if the virus could exist without it.
Absolutely.
Is it actually complexing with the genome?
We don't think it's actually complexing with the genome when we do these non-denaturing gels. So sometimes we see some association of these RNAs with genome, but what happens is basically any two RNAs that you put in a virion, we'll see them together again by those charges that I talked to you about RNA, just by the condensation of RNAs and their particles. But we do know that those RNAs are sort of in the region that they could be used by reverse transcription, because another fun thing that we've done sometimes is we put in sort of like decoys that can grab onto reverse transcription intermediates, and they will kill the virus. So clearly they're there, and the virus can jump onto them.
So the genome requires a signal to get into the capsid. How do these get in?
So Sarah's been mapping these sorts of things, and with the 7SL we have a pretty good feeling now, and it's a pretty interesting and complex dance that there's interactions of different parts of the RNA with different parts of the GAG polyprotein, at least genetically, and then she can map portions of that 7SL that seem to be critical. And motion is also necessary, I would say, because we can lock the RNA into shapes that are not flexible, that people did in the past to solve RNA structures, and they are not incorporated
Does
anymore.
It look anything like the size sequence of the virus?
No. And in fact, it doesn't compete with the size sequence of the virus. So this is, in fact, I guess this would bring us back to part of the reason why I did the project that I did for PEI initially, is we discovered, if you will.
Who paid you, by the way?
My collaborator. I mean, he got a subcontract for us.
Okay.
From the grant,
basically.
right.
Yeah.
So we have a subcontract to his NIH grant. It's sometimes hard to quantify retroviruses. You'd think it would be quite easy because they have RT and they have proteins and so forth. But there's a lot of irreproducibility in a lot of my colleagues in the field's work. And because we discovered that these RNAs were packaged in proportion to GAG, regardless of whether or not the genome was there, we will normalize to viral proteins, but we find it much more precise to normalize the 7SL concentration in virions. So in fact, it was a poster at a meeting that I was telling this to my collaborator that he first asked me to collaborate with him.
So these aren't small RNAs you found in Maloney. You've done this in Maloney. Does HIV do
so
the same thing?
So 7SL RNA is in all retroviruses.
Okay.
And there's some kind of fun genetics that we have that actually, with some other viral systems, where we can make some viruses that do and don't have them. So we do have some sense of viral proteins that are involved in recruitment of some of the others.
It's great. Sounds really neat. This is what you're excited about right now.
Well, yes. I mean, and I guess then the point would be, of course, where we're taking it from there. And I think that and the other stuff is genetic recombination, but that's for another day.
Okay. Well, some viruses don't have packaging sequences, like the one I work on.
So how does it recruit its genome?
And it is specific. Nothing else gets into the coronavirus virions, but
so is it location?
Maybe.
Because, I mean, that's the other thing that's very cool in our virus, is that we think we know how two RNAs come together and kiss and dimerize. And
how do you know it doesn't have a packaging sequence?
Because people have looked and they never found it.
But sometimes these things are not as straightforward as you think, because there may be, for example, the fact that some region is not translated at a certain time opens up something to be bound by, or spliced in a certain way, and it's marked by a factor that recruits it or takes it to a certain place.
I mean, at least the straightforward ways of identifying packaging sequences that have worked in other viruses have not. And so it's something unusual. I mean, I'm sure there's some.
But you say it's straightforward, and yet, as you pointed out earlier, I mean, a lot of people study retroviruses, and it's taken 30 years, so it's not as straightforward.
So this SRP RNA is normally in an RNP complex.
It is.
That correct? Is that does HIV have to kind of is it ever free in the cell, or is HIV?
So in fact, these are the other things that I guess I will tell an intersection of the story of the YRNA that I told you. Another reason why we studied that one and the 7SL. Remarkably, we don't see any SRP proteins in virions, and so this is very old work from Peter Walter, that you would say that all the proteins. So some of the proteins start RNP assembly in the nucleus, maybe even co-transcriptionally. Others join in the cytoplasm. We see none of those in virions. So the question then is, are they stripped off, or were they never there to start with? And in fact, there were several reasons why we think they were never there to start with. One of them, is sort of related to this YRNA that I told you, that it gets recruited even though it's not present in the cell. So it must have been picked up right early on when it wasn't decorated with proteins, right? But in the case also then of the 7SL, Sarah in my lab now has mutagenized a lot of the protein binding sites, and it still is packaged. And in fact, sometimes she even sees less of the RNAs in the cell, probably because they're not so stable anymore, but they still are packaged quite well.
And so this is all telling. Are you thinking that there's this RNA gag interaction going on in the nucleus?
So this is something that Leslie Parent has shown in the, and I actually, it's so interesting because I've been talking about this a lot over the last week or two, and there have been about 8 different groups of scientists who've come up to me and said, we think that now too, but they're not going to say it, or they think somebody else reviewed their paper, and I won't name names and stuff, but certainly this is something that Leslie Parent has seen in RSV. And then there have been the last 10 or 12 years that basically if you suggested that happened, that your paper would never be accepted, your grant would be triaged and you would be laughed at. And now, as I say, many people are coming up and sort of whispering to me. I mean, many people, So we'll have to see how this goes, but I think that one of the things that's interesting is that some of my advanced imaging friends then now say things like, the techniques that we have, you would not be able to detect those above background. And we're sort of postulating that these are transient interactions. And I still actually am not as convinced as some of these other guys, because I think other possibilities are out there. Again, that there may be minor factors that interact with these RNAs that may be pulling them in certain directions that we don't appreciate. In fact, actually, some of the minor proteins that another collaborator of ours has identified as binding to the tiny, tiny bit of RNA that's left in the knockout cells. Some of those RNAs have been implicated as possible restriction factors in retroviral replication too. So there's many different ways that we might be thinking about this.
Nice, good story. All right, let's do a few other items here on TWiV We usually do some email, which I want to do. Before we do that, Alice sent me a story this week that I want to tell you all. She sent me an email. The subject was virologist tapped to lead Apple. You may know that Steve Jobs passed away recently. He was the chairman of the board of Apple. He was replaced by Art Levinson, who's a virologist. So how about that? A virologist is now running Apple. So I posted this on our TWiV Facebook page and got a bunch of really cool responses. And one of them was, I thought Apple computers didn't get viruses. And then someone else, I said, he probably never listens to TWiV. And someone said, if he has never listened to TWiV, then Apple is going down. And finally, someone said, he should be going to Microsoft. They have the viruses. So thanks for that story. That was pretty funny. We'll put a link to that. So Art Levinson worked on adenovirus as a PhD student. He worked on retroviruses with Varmish and Bishop. He has 35 virology papers. Pretty impressive. So virologists in high places. That's pretty cool. Now, Ricardo sent me, who's a listener in Portugal, sent me a link to an article which apparently indicates that on some Delta flights, they are running anti-vaccine informational movies. Anybody see it? Had been on a flight lately? Yeah. So the American Academy of Pediatrics wrote a letter to Delta to object to this, and Delta said, "No, we're not going to stop showing them. We could show whatever we want on our planes, basically." So this is a video saying you shouldn't get the flu vaccine. It's not necessary, essentially. And the video was online, but I think it's gone now. So I posted this on Google+, and someone immediately came on and replied, "Your unquestioning advocacy of all vaccines is as bad as the opposition to all vaccines. Surely you must see that big pharma financial interests are often a greater driving force than science." So this is what we often see on TWiV. We have a number of anti-vaccine people who somehow feel that big pharma and profit override science in the use of vaccines. And so my comment is, of course, that we're always critical of vaccines that aren't good. We have talked about those on the show. And if you go out into the world and be scientists, this is going to be part of your job to counter this notion that many people have, that vaccines are not necessary, that they're bad, and they're just a way for big pharma to make money. It's a big problem. So that was from Ricardo. Our next email is from Ken, who writes, "One of your emails included the request for us non-academics. I'd love to hear a day in the life episode of a graduate student, a postdoc, a PI, and so on. It reminded me that in 1999, journalist John Franklin wrote a long piece for the Raleigh New Observer to make a mouse that did a great job conveying the daily life of a senior graduate student, especially the degree to which progress involves failed experiment after failed experiment. Unfortunately, this was back when newspapers didn't leave stories on the web for long, so it seems the piece is only available as a document file from the author. And so we have a link to this file so we can take a look. To Make a Mouse, John Franklin. So thanks for that. And we plan to have people on future TWiVs to talk about a day in the life. That would be a lot of fun. The next one is from Jen, who says, and this is for Alice and Kathy too, "Hey, Vince and friends, guess I should start off by saying how much I love your podcast. I discovered TWiV shortly after falling in love with viruses in my first microbiology class and have been addicted to them ever since. I was surprised to discover that such an academic subject which would have its own podcast. Anyways, I'm currently a community college student and plan to transfer to a 4-year university next fall. I've been planning on majoring in biochemistry after transfer, but that was before I discovered how fascinating virology is. My question, assuming I want to get a graduate degree in a virology-related field, should I keep my current biochemistry major or should I major in microbiology, molecular biology? I'm sorry to bug you with such a trivial question, but I don't know anyone else who would have any insight into this subject, and I really want to maximize my chances of getting in to study something I love, viruses. Any reply would be appreciated. Keep up the awesome podcasts."
That's not a trivial question. That's not a trivial question at all. I guess my first answer is do what you want to do. Okay, I think that's the most important thing. But I would certainly not discourage somebody, especially at the undergraduate level, from pursuing biochemistry. And I think not only would it not be a bad thing in terms of foundation for virology, it's a good thing. To me, biochemistry is the language that we speak, okay? And if you don't understand the biochemistry, you're missing a lot. A lot of people can talk like they know what's going on, but then if you peel back the cover and get down there, you discover there's no biochemistry there, so they don't really understand what's going on. All right, I think biochemistry is absolutely critical. And so if you can come in with a foundation in biochemistry, I think that's really important. So I'd use the opportunity.
Kathy?
I think majoring in biochemistry is fine, and probably the major has room for an elective, and she could take more microbiology that way. And then I also think, too, about, any of our disciplines. Rich just made a pitch for biochemistry, but I would make a pitch for genetics. And so, in fact, when I was first applying for jobs, I applied for jobs in a variety of types of departments: microbiology, genetics, biochemistry. And so in the same way, a student coming in with a background from any of those fields should be able to do okay. And if she has the chance to try and do some undergraduate research in a microbiology lab or a biochemistry lab, that would put her in a good position for graduate school.
What do you think?
I agree, but I think obviously as hardcore of a science as you want to do, I think is good, and maybe biochemistry is a little bit more fundamental. I think virology builds on. I never took a virology class. I had to learn it. I actually then did get the notes from Hamish Young and Vince Racaniello here, and I taught off their classes for a few years till I started understanding what it was.
Yeah, I never really had a real biochemistry class. I took molecular biology. I never took real hardcore biochemistry.
So that's why he thinks you should do it, because he still doesn't understand it.
Yeah, right. And my first job was in a biochemistry department. I've never taken a microbiology course. And now I'm in a microbiology department.
Yeah, I'm a professor of micro and immuno, and I still have not taken immunology. Frightens me.
Well, I was a biology major. I didn't take any virology courses in college. I didn't take biochemistry, just organic and all the other stuff. So I turned out to be a virologist as well. So I think that you do, as Rich said, you do what you really enjoy. And if it's biochem, that's great. I don't know any virology program that would not want a biochem.
Absolutely.
So that's basically.
I think you need it to get into graduate school. So
yeah. All right, the next one is from Don. Right site. Vincent, Dixon, Allen, and Rich, I hope you will allow me to address you by your first names. After all the hours I have spent with you, I feel that I know you. I'm a longtime listener and believe that your podcast has made my life richer in ways that would be difficult to describe.
And Eleanor and Vincent.
That's right. Difficult to describe.
I don't want to know. I don't want to know.
There is one thing that bothers me, however, and I hope you will indulge me for a moment. It seems that you have a bias towards accepting research at face value without an appreciation that some research is skewed by scientists and drug companies for monetary or other considerations. Please don't take this as an aspersion or insult to the character of anyone on the podcast. In a word, I guess that skepticism is what I see as sometimes missing. Anyway, I don't think this email needs to be read online.
Too late.
Sorry. But I would like to recommend a pick of the week. The lesson is, if you write us, we'll read
it gets read, yeah.
A pick of the week, we'll save his pick of the week for later. That has something to do with skepticism, and I'll read this later.
And it's a good pick. We'll do that later.
I might forget. Can you remind me, Rich?
No,
Okay.
I will forget, too.
So,
okay? But no, actually, I'll put that in my picks somewhere.
I think we are skeptical often.
Mm-hmm.
I mean, those of you who have listened, do you think we accept everything at face value? No, a lot of shaking no heads.
So there was we did a significant part of a TWiV on this. We did a paper called "The Decline Effect and the Scientific Method."
That's right.
Which is all about this. That's TWiV 123, and then look at the whole years-long saga with XMRV. We chased that around with nothing but skepticism.
Quite.
And
And we got a lot of criticism for being skeptical of that,
right.
if you recall.
But I think the other thing that may give the appearance of not being skeptical is that we also make an effort to pick out really good papers. All right? And so if you're doing really good papers all the time, although we probe underneath those, they're good enough so that you're not going to be very skeptical in the end. You're going to be pretty positive about the outcome. But he's right. Skepticism is really important.
Yeah, I agree.
He's right that there are some people who will use science in nefarious ways. Okay, so it's a good point, and skepticism is really important.
Do you think we should, from time to time, do a paper that we don't believe?
Absolutely.
You think?
Yeah.
I mean, my view is there's so much good stuff that we don't even have time for that. So we want to showcase virology, right? But maybe just to illustrate what constitutes not such a good paper.
Actually, if we look back if we keep this in mind and look back at the episodes, or even look forward, we're probably going to, if we keep it in mind, we'll find plenty of incidents where stuff comes up that we're skeptical about, and we can, point that out.
And I want to point, you just mentioned the XMRV, and just because by chance I was listening to the Steve Goff's first episode about XMRV, and if you listen to it now, it's very illuminating that he keeps saying, this is really unbelievable, and this just doesn't seem right. And but the way he was saying was with skepticism, which ended up being based in a lifetime in the field, and the history bore out, so it was interesting.
Well, and in fact, in retrospect now, this 2009 science paper describing the association of XMRV and CFS that turns out to be mostly wrong. So there's an example of not a very good paper that has...
True.
And so there it is. So
an apparently very good paper.
Yeah, so we all thought it was. So this is an issue, as you can tell, twice now we've mentioned this conflict between selling things and making money. And I agree that it's a problem. In the best world, all drugs and vaccines would be free. But we do live in a capitalist, free market country. And the reality is businesses have to make money. Otherwise, it's not a capitalist society. So I don't know how you do science any other way. But point is well taken. All right, let's do one more email. This one is from James. Thank you for filling my bicycling and running time with virology discussions, which are excellently informative as well as enjoyable, a combination not always to be expected from podcasts. I know that you are working on a grant proposal to generate what I imagine is much-needed funding. Are we? Not just for TWiV, right? I have a suggestion which might generate a small amount of discretionary funds as well as provide some verifiable data for consideration by your advertisers. I subscribe, actually pay for several podcasts which provide additional material to be downloaded automatically in iTunes. I find it difficult to believe that you would not have 15 minutes of additional discussion already recorded for each podcast that could be supplied as extra listening for subscribers. The extra sessions could be, 1, an additional paper that you could not fit in when in journal club mode, 2, a short interview with an outside expert who did not have time to participate in the entire TWiV recording, 3, an informal discussion of points you did not think or have time to bring up during the regular discussions, 4, a breaking news item that went to press after recording the podcast but was of sufficient interest to not wait for the next week, or 5, shorter versions of your Virology 101 discussions, which might be easier to produce than a full hour. I would think that $2 per month for an annual total of $25, which seems reasonable to me, would be acceptable to others. Thank you for all the instruction, laughs, insight into other topics, e.g. vertical farms, and just plain fun you bring to my week. You are even managing to teach some virology to one who never had time while in school. So James is a PhD, a toxicologist in the Office of Human and Ecological Risk in California. So those are some good ideas.
Yep.
I just don't like to charge people at all.
You know, there's another thing we could do, and that is, put an opportunity on the website to actually donate. And just make a volunteer.
Good to put a coffee mug. Buy us a cup of coffee, sure.
But then you probably need to become a 503(c).
I don't want to have. We have to do something so that money goes to the podcast, not to me.
Right.
I don't want that exactly. Okay, so that'll do it for our emails for today. Let's do a couple of picks of the week. Alice, what do you have for us?
Yeah, it's just there were so many possibilities, but in the end, I guess I know that my student Sarah says she likes books, so I chose a book, and this book is called Banvard's Folly: 13 Tales of People Who Didn't Change the World. And in some ways, maybe this ties in with some of the discussion we were having earlier. So the back tells you about the fact that in Banvard's Folly, Paul Collins celebrates what he calls the forgotten ephemera of genius. Here are 13 unforgettable people who claimed... So basically what happens is that these people were hugely famous in their time for scientific discoveries that we've totally forgotten, mostly because they weren't right, like N-rays. Has anyone heard about N-rays, for example? And then some of these things are, they are people who were fabulously popular authors whose work is now totally not known, but, contemporaries of Walt Whitman, and nobody would have heard of Walt Whitman at the time because this other person was so famous or whatever like this, and people who were charlatans in some cases. So I think it's quite fun. I picked this book up about 6 years ago when I was having to deal with a person who was a person who bought into the vaccine and autism thing and who was very into things like chelation and flashing lights and all this sort of stuff. And I kind of was interested in the whole issue of that kind of aspect of science. And I think it's so I guess I like this book because I come back to it in my mind thinking about how these sorts of things have happened before, and they've resolved, as you pointed out, and we now know Walt Whitman instead of whoever the author was in this book. So that's my pick. I just found it by looking for that kind of stuff, and there it is.
Thanks very much. Kathy, what do you have for us?
So several weeks ago around Halloween, you probably heard news stories about how the Earth's population was going to surpass 7 billion. And I heard on BBC that there was this website that you could go to, and you can find out what your place was, or what person you are in the count of the world. And I'll just round it off for me. So you put in your birthday, and I'm the 2.7 billionth person alive on Earth. Then you can also find out where you stand in history. So I'm the 76 billionth person to have lived on Earth since history began. And they show you a population of the world and you are here kind of thing. And then you can click on several other pages. You can also link out to a UN Population Fund site. But it shows you the current count of people in the world, the fastest growing country, the fastest shrinking country. You can put in your own country and find out the current population and yearly population growth. And then you put in whether you're male or female, and it calculates for you your life expectancy, and it summarizes everything for you. So it's kind of a fun way to figure out your number on the Earth.
Mine was close to yours.
Yeah, I figured out you were born the same year as the DNA structure. Yeah.
Yeah, I did it, and I don't have long to live.
Well, 6,400 deaths every hour. This is great. I hadn't seen this.
That's great.
There are 15,000 births also. So we're in positive population growth. That's great. Thank you, Kathy. Rich, what do you have for us?
Okay, so I'm really going to push the envelope of the definition of science picks. Have any of you seen the movie The Princess Bride? A lot of nods. Let's see hands. How many hands? All right. Almost. So how does this fit as a science pick? Two reasons. One, there's this wonderful exchange between Vizzini and the Dread Pirate Roberts that to me is a study in rationality, where Vizzini's going through all of his logical arguments about which cup holds the iocane powder, and it's just ridiculous. It's too long to relate here. You have to see the movie. But the other one I really love is when they take Wesley to Miracle Max, the doctor, okay, because they think he's dead, and Miracle Max revives him. Miracle Max is played by Billy Crystal, and he says, "Woohoo, look who knows so much. It just so happens that your friend here is only mostly dead. There's a big difference between mostly dead and all dead. Mostly dead is slightly alive." And then he revives him. I can't tell you how many times I've used that in class, mostly with respect to leaky TS mutants, right? I say, it's only mostly dead. It's mostly dead is not all dead, it's still slightly alive. So that's my reason, but beyond that, everybody needs to see The Princess Bride.
It seems kind of a stretch, but
It is a stretch. I told you it was a stretch, but I needed to publicize The Princess Bride because everybody needs to see it.
Is that how you view viruses, as mostly dead or somewhat alive?
Oh, no, they're alive.
Yeah, okay. All right, that's for another time.
No, actually, I like the definition that we've come up with is that they're undead.
Right, they're zombies, right?
Right.
Yeah.
Undead.
Viruses are zombies. Okay. My pick is a website at the ASM called the Center for the History of Microbiology. So I get monthly emails from ASM, and this one came recently, and they have an archive of various items relating to the history of microbiology. They have bibliographies, documents, book collections, visual aids. Nice stuff if you're teaching and you need some chronicle of the history. So check that out. We'll put the link to that in the show notes. And let's do our listener picks of the week. That's right. We have two today. One is from Tim. One of my friends and colleagues here in Cincinnati, Dick Ward, is now mostly retired, but recently penned an autobiography about creating the rotavirus vaccine. The book is called Dead Ends to Somewhere and can be found at this link. It might be of interest to you and your listeners. While the majority of the book is about his personal struggles, and only near the end does he discuss Rotarix, in telling his story, he conveys the trials and tribulations of young scientists trying to find their way and establish a niche and a purpose. It is also a lesson in serendipity in science, as his experience in viruses such as reovirus, which at the time seemed to be for naught, ultimately served him well when he found himself working with rotavirus. I had lunch with him last week and told him about TWiV, so I might have found you and other listeners. So Tim is a professor of pediatrics in Cincinnati, who is a listener and who has also started his own podcast. He's inspired by TWiV. It's called This Week in Pediatric Oncology. So Timothy is an oncologist, and specifically a pediatric oncologist. So that is one listener pick of the week. And then the other one is from our listener who wanted more skepticism. He recommends this pick of the week that expresses my skepticism more eloquently than I am capable of doing. It's a TED Talk by Ben Goldacre titled Battling Bad Science. You probably know Ben Goldacre from the UK, who is a big skeptic himself. So he's sending us a link to that video.
I watched that video. It's actually quite good. It's good. It's good. Worth looking at.
So thanks for that. So this is TWiV, which we do every Friday. We release it on Sunday on iTunes or the Zune Marketplace. You can subscribe there. And if you're new to TWiV, please subscribe and write a brief review at the iTunes page. That helps to keep us on the front page, which we are often on. And that way we get more listeners. We also have an app that you can use to stream the episodes to your smartphone, either an iPhone or an Android device. That's at microbeworld.org/app. You can also find us at twiv.tv, where we keep our show notes and letters. And of course, don't forget our Facebook fan page, facebook.com/thisweekinvirology. Send us your questions and comments to twiv@twiv.tv. Kathy Spindler, thank you for joining us today.
Thanks for having me, and thanks for coming.
You're welcome. Kathy, of course, is here at the University of Michigan Medical School, as is Alice Telesnitsky. Thank you for coming.
Thank you. It's been great.
I hope your kids enjoy it.
I hope so, too. You know, they may change their majors, huh?
Right. Rich Condit, University of Florida, Gainesville, where it's warm and sunny.
Right.
Always
a good time.
Yep,
it's great to be here.
And I'm Vincent Racaniello. You can find me at virology.ws. I want to thank the postdocs in microbiology and immunology for inviting me to come here, particularly Stefan Taube for taking care of me, and to Kathy for inviting Rich and thinking, well, maybe we should get them together and do a TWiV. So it's really been great to be here. I want to thank Matt and Marilyn for doing the audio today. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.